Oops, sorry about that. Hello, everyone. I'm Madhu, working as a postdoc at the University of Ottawa, Canada. I am a computational biologist. Right now, I turn into a bioinformatician. I work on the arbuscular mycorrhizal fungi, and this is like a first time I'm working on like this fungi. And I'm going to present my recent research, which has been published about the early branching genomes, that is Paraglomus and the geosiphon. So those genomes. The arbuscular mycorrhizal fungus are the prominent symbiont, which forms the symbiosis with the almost 70% of the land plants. And also it is phylogenetically placed in the Glomeromycetina clade. And this species, so in the whole aim of clade, only the Rhizophagous irregularis is studied. Rest of them are like not studied well. So the early lineages such as Archaeospirales and the Paraglomus are understudied because there is like a difficulties in extracting the DNA because it exists in the natural environment and the cultures are present in the pot culture. So it is difficult to, extract the sample. And there were like no genome reports available. I published after that. So these are the questions I try to address on my papers. So it's possible to assemble the contaminant-free genome from the pod culture, and did the losses linked with the obligate biotrophy or occurs in AMF, or all the AMF genomes are large and full of repeats. So this is like one of the interesting AMF. This is the geosiphon piriformis, where it forms a symbiosis with the bacteria, whereas sorry. Already I was nervous and this is making me more nervous. So yeah, geosiphon, it is one of the only one AMF which forms symbiosis with the cyanobacteria. And this very unique about this geosiphon is it have a bladder like a structure. So whereas that bladder contains the Nostoc cells inside that bladder. And this is like a pictorial representation where you can see the Nostoc cells are beautifully arranged inside the bladder. And this species is originally found in Germany. So this was initially thought to be lost, but my collaborator from Germany, she managed to find this species from the Spessart Mountains, and that's how this project started. And she contacted, she was like, yeah, I have a bioinformatician to work on, and my supervisor asked me to work on this. That's how, yeah. And this symbiosis was like one of the oldest ancestral symbiosis in the AM fungi. So if you look into this data, you can see there are like a, there's a bimodal distribution from the sequence data. It is because like this sequencing breeds are coming out from the whole breeds, from the soil and everything. It contains like all other organisms. So it is, this is how it looked before, filtering and everything. Then I use the filtering, then I use a specific method, whereas I use this approach for the both geosiphon and the paraglomus genome. From the genomic reads, I tried to assemble the metagenomic context. And from those metagenomic contexts, what I did was like I clustered those clusters on the basis of the tetranucleotide frequency and also on the coverage. So on the basis of the nucleotide frequency, which is like conserved, so I separated those clusters, then I aligned those clusters, like whichever is like similar to AMF, I took those clusters and I tried to perform the genomic assembly. And once I had the cluster, from there, I extracted the reads and I mapped back to the reads of the whole reads. Then I extracted, then again, it was like a painful job to do multiple steps. So after filtering, this is how it looked like. So yeah. And after getting the genomic data and everything, so we were interested to see the comparative genomic aspects and also to see what is like so unique and special about this like early diverging fungi. So if you look into the genomic stat, you can see this paraglomus and geosiphon. It have unique things such as, like for a paraglomus, you can see the size of the genome is like 44 MB, whereas rest of the arbuscular mycorrhizal fungus ranges from 100 to 150 MB. So this is one of the smallest AM genome which has been like found so far. And if you see the repeat content, and the repeat content is also very less, where the AM fungi are known to have more repeats, whereas this genome contains less repeats. And the gene density and the GC content is also high in comparison to all other AMF. And the highlighted ones are AM fungi, and rest of them are like outgroups from the Mucormycotina. And you can see in the geosiphon too. So like the genome size of the geosiphon is 129 MB. And yeah, so the repeat content is also much higher. It's like 64% of the genome contain repeats. So, you know, this is more repetitive than we thought. And yeah, and also like the paraglomus, it had a fewer gene models, like around only 10,000 gene models, whereas all other AMF contains like more than 20,000 gene models. And with the collaboration, we tried to do the phylogenetic study to see where this geosiphon and the paraglomus sits. And we found out that this geosiphon and the paraglomus is the, they both are sister lineages. And the paraglomus is the sister to the ambispora and the geosiphon. And this AMF fungi is not known to have the plant cell wall degrading enzyme. These enzymes are originally absent in all other AMF fungi. So we wanted to know whether these plant cell wall degrading enzymes are present in geosiphon and also in the paraglomus. We saw that these, these plant cell wall degrading enzymes are not present also in the early diverging fungi. So that means it's like completely lost, and it was not having those enzymes since the origin. And we try to see like domain family expansions, and whereas which is like which occurs in all other AM fungi too. We saw these repeat related domains such as BTP, CEL1, and WD40 domains, which are expanded in all the AMF. And also these expansions are also present in the geosiphon and also with the paraglomus. So these AMF are known to have like more transposon. So I compared the transposon with all the AM fungi, and we saw that like very few T elements in the paraglomus. And another thing is like there is an expansion of the gypsy elements in the geosiphon in comparison to all of the AMF, whereas gigaspora is like a 500 MB genome, which contains like 8,000 some gypsy content, whereas geosiphon is like 120 MB. It contains like just more than that genome. And also we saw that I don't know. I didn't do anything. Yes, we are back. So, and I compared the genome size and the repeat content in all the AMF fungi data which we have, and it says like the genome size is correlating with the repeat content. It is very much significant. So, and we performed the core gene analysis with the early diverging fungi with the other early diverging AMF with all other AMF. And also we included the Mucormycotina genomes. And I found out that like in the Paraglomus, it contained the more core gene in comparison to all other genome. And also the 39%, it contained the more species-specific genes in comparison to all other AM fungi. And also we found out that there is like it might be possible that the Paraglomus have gone under the species-specific genomic reduction. And another thing is like important thing is like we found out that the geosiphon had more expansion of the species-specific genes, and we found out that the lipase 3, particularly the lipase 3 domain, has been expanded in the geosiphon genome. So it might be possible that these lipases are like, transferred from the cyanobacteria called nostoc. And I didn't include that result here. We saw an evidence that these lipases are transferred horizontally to the geosiphon. And we looked into the mating locus, and we saw that the mat locus is conserved in all the AMF, and it is also like conserved in the ancestral clade too. So, and we also found an evidence for the meiosis specific genes too. So these are the summary key points. Kamer-based approaches worked well with the pod culture, and also the phylogenetic placement suggests that this early diverging genome is the sister clade of the geosiphon and the ambispora. And most the MRCA-specific genes in the Glomeromycotina are originally lost, such as the genes involved in the fatty acid and the thymine metabolism. And all the Glomeromycotina genes contain the meiosis-specific genes and also the mat locus. And the gene and the repeat content can vary significantly with the Glomeromycotina and correlate with the genome size. And the streamlined genome of the Paraglomus may reflect there might be a species adaptive process that indicate the MRCA of the Glomeromycotina may carry a small and gene-rich and gene-poor genome. And these are my acknowledgement. I'm extremely thankful to my lab mates who taught me fungi, mat locus, etc., and my collaborators and my PI. Without my collaborators, it would not been possible to work on the geosiphon and anything. Thank you.
Thank you, and I'm very sorry for the technical trouble. So, any questions, please? Okay, people are, please.
So genome size and some of these other genomic properties, they just look superficially related to spore size. Mm-hmm. So is there any way to kind of predict that with some of these spore traits?
With the bioinformatically, if you have a sequencing reads, yes, we can estimate the genome sequencing, genome size. So based on the Kamer coverage, so we can calculate like how much the number of data is there, and from there we can calculate. There's a formula too. I can give you.
There is a question online. What is the significance of repeats?
Repeats, I mean, maybe it is helping these genomes to, get genes from other endosymbionts, or we don't know, maybe it plays a role in the adaptation or anything.
Great talk, and really moving kind of things for the field of AM fungi. So that's super awesome and exciting. My question is related to the plant cell plant cell wall degrading enzymes. And you said how Paraglomus might not have that in its genome. How do you think then it will infiltrate? How has it evolved infiltrating the root cell then?
I'm not sure, but it's been like all the other AMF doesn't have this plant cell wall degrading enzyme. So it seems like the plant cell is like just, somehow letting the AM fungi to enter into the plant cell. So that's all I know.
Thank you for the nice talk. And I have a question about the technical term. And in presentation you mentioned you determine the genome sequence of the Paraglomus species from the pot culture. So my question is, how did you eliminate the possibility of the contamination of the other microorganisms from, for example, like their surface?
So
compared to the pure culture. Sorry.
So I mean, like the main aim of the project was to get the just Paraglomus. So I calculated the tetranucleotide frequency, which is like conserved in every organism. So whenever there is like a tetranucleotide frequency, which is similar to particularly to the Paraglomus, it separates that particular cluster to rest of the other microbiomes. So that's how I separated like other organism and with this. So now there is like a follow-up project going on to study other endosymbionts which are associated with the Paraglomus and geosiphon. So it's going to be exciting.
And what are the big picture implications of your findings?
Future implication? Okay, so it is now geosiphon can form the symbiosis with the cyanobacteria. So why not Rhizophagus irregularis? So people can work on that and to know whether it can form the symbiosis. So.
Okay, we can have one more question. Okay, I will ask.
Yeah.
So how would you compare the gene content of those early lineages in Paraglomus and geosiphon and glomerales?
So take the gene models and compare it with the sequentially, sequence-based searches and sequence similarity. So that's how I compared it.
Yeah, but do you find more than expected amounts of similarities, or are there too few or less than expected in the content of different genes?
In the content of different genes. So, I mean, I am not getting it fully.
Okay, never mind. So thank you again.
Thank you very much.
And let's thank all speakers for this session.